First of all, I'd like to thank the organizers for giving me the opportunity to present our work to you today. My talk is a little bit different. We don't have heterochromatin data, but we are going to stay with the overall theme of rejuvenation. And it's great that I'm placed in this position in the series because we are going to be talking a little bit about muscle, a little bit about brain, a little bit about both. So as you can see, I have altered the title of my talk a little bit just to reflect kind of the take-home message that I want to leave you with today is that we study this new muscle-to-brain signaling axis that we believe underlies many of the brain benefits of exercise. So this is an exercise talk where there's actually no exercise. So spoilers. So I realized just now that I should have maybe changed the slide to benchmarks of the aging brain after our awesome session yesterday. But the aging brain, just like all other aging tissues, shares a variety of hallmarks or biomarkers. And today we're going to be talking about 3 that have actually been mentioned in previous sessions yesterday and today. So the aging brain is characterized, as most other aging tissues, by the accumulation of protein inclusions and protein aggregates, which are shown there, which is associated with a decline in proteostasis function. As Sun just beautifully introduced to us the concept of neurogenesis in the brain, where there is a decline both in the number of neural stem cells, but also in their ability to divide, mature, and integrate into existing networks. And finally, in the aging brain, there's an overall increase in neuroinflammation, which is the chronic activation of astrocytes and microglia towards pro-inflammatory phenotypes. And overall, we understand that these are central drivers of the process of brain aging and contribute to classical phenotypes that we see in the aging brain, which is the thinning of the cortical areas and the expansion of the ventricles. So in addition to that, these mechanisms are also shared with neurodegenerative diseases and are believed to contribute as well to the increased risk for the development of these disorders in the aging brain. So I don't think I need to tell this audience that one of the most powerful interventions we have against the development of these phenotypes is exercise. However, to date, the mechanisms that underlie the benefits of exercise in the aging brain remain poorly understood. Mechanistic work in animal models has revealed that most of them appear to be mediated through increased levels of brain-derived neurotrophic factor in the hippocampus. And so it is believed that this BDNF expression may be able to essentially oppose this decline in proteostasis, decline in neurogenesis, and increases in inflammation. So in the field of exercise in the past decade or so, we've come to understand that during what I like to call behavioral exercise, when we would go running, when we get on the treadmill, when we go dancing or swimming, a lot of our peripheral organs, and in this cartoon, I'm highlighting skeletal muscle and liver, secrete a myriad of circulating factors into the blood. And these factors move through our circulatory system and through mechanisms that we still don't quite understand. They hit the blood-brain barrier or cross the blood-brain barrier to drive the activation of this hippocampal BDNF expression, and ultimately leading to central nervous system rejuvenation. And there's intense study in this area. And we see things like increases in angiogenesis in the brain, reactivation of adult neurogenesis, preservation of synaptic plasticity, and improved cognitive ability. And so the existence of these exercise-induced secreted factors has opened up a window of intervention for us. And this is the space that my lab currently lives in, where we're trying to see is essentially characterize and phenotype these markers and see if we can use them to develop what I like to call exercise mimicking therapeutics, essentially drugs that can activate these factors in circulation and activate the CNS rejuvenation mechanisms in the aging brain. So we've been talking about exercise. What does exercise do? Well, many things, but let's focus on its effects in skeletal muscle, which is essential for actually exercising. And so in rodents, voluntary exercise is a well-studied mechanism of exercise activation. And in skeletal muscle, what we've come to know through decades of research is that running in particular can oppose pretty much every hallmark of aging that you look at in skeletal muscle. You can see that here. It promotes beneficial metabolic reprogramming, and importantly, it actually reactivates proteostasis, both autophagy and the proteasome, and it has powerful effects on mitophagy, so the recycling of mitochondria, which you can imagine is essential for energy production during exercise. And it is believed to be that kind of this timeline is what drives essentially the metabolic health associated with exercise. But I was really interested in kind of this area down here. My favorite hallmark of aging is proteostasis, by the way. I think we were taking votes yesterday. And so in particular, the way I study autophagy is through the use of this factor, transcription factor EB, or TFEB for short, which is a master regulator of lysosomal function in autophagy. And to make, beautiful work summarized in one slide, essentially it's a transcription factor that drives the activation of multiple steps of the autophagy and lysosomal function pathway, making it to date the most powerful activator, both in vivo and in vitro for autophagy. And so I'm a mouse modeler, so I made an inducible human TFEB transgenic mouse by placing the human TFEB transgene downstream of a stop codon, such that there would only be TFEB expression in the presence of a Cre recombinase enzyme that would flox out this entire cassette. And so to drive expression of TFEB to skeletal muscle only, I crossed these floxSTOP-TFEB transgenic mice with a human skeletal actin Cre, HSA-Cre, which is a well-characterized line for skeletal muscle specific expression. And these are the animals that I'm going to be telling you about for the rest of the talk. So first, here's confirmation that we indeed can achieve expression of our 3X flag tagged human TFEB transgene. These are skeletal muscle immunoblots of double transgenic individuals. You can see expression of TFEB right there that is not present in the controls. And this is specific to muscle. We've done extensive characterization, but for the sake of time, today I'm only showing you an immunoblot of the hemibrain immunoblot lysates of the same individuals. You can see a skeletal muscle positive sample here. There is no TFEB expression in the brain, but you can see the flag AGFP, which is this fragment right here before the stop codon, to confirm genotype identity. And so our first question was, what is happening in skeletal muscle by activation of TFEB throughout the lifespan of these animals? As we know, proteostasis goes down, TFEB activates proteostasis. Are we correcting or changing some hallmarks of aging? And so there's a lot of work here that I'm going to skip over in the interest of time, but essentially we did proteomic studies of skeletal muscle at a young time point, 6 months of age, and an age time point, 18 months of age. And as you can see, there are many, many things that are changing in the proteome landscape of TFEB expressing skeletal muscle. And TFEB, you can see, is one of the most differentially overexpressed protein. And so what does this mean functionally? So we did functional pathway analysis, and we were very excited to see things that are hallmark of aging related. And so we see pathways that change that are proteostasis associated. A large number of hits are hitting mitochondrial metabolism. There's interestingly a big cluster of fatty acid metabolism pathways. Some age-associated signaling was differentially expressed, and then also a big hit on amino acid metabolism. So I very much enjoyed the session yesterday talking about branched chain amino acids, because that pathway is heavily modified in these mice. And so again, to summarize, a lot of work being done by one of my grad students at UAB. We are starting to characterize things that change in the muscle of these animals that we believe validate our hypothesis that we have essentially created an exercise mimicking state. And so the skeletal muscle adaptations that we see in TFEB expressing skeletal muscle are similar to those that are seen after endurance exercise. The muscle of these mice is not hypertrophic, it's about the same size. There is a change in fiber type switch, and this has to do with changes in mitochondria amount and as well as type of mitochondria. We're still characterizing that. But there is definitely increased proteostasis. We've done extensive studies on that. There's increased mitochondrial biogenesis and function. There's increased capillary density and blood flow to the muscle of these animals, increased oxidative capacity. reduced hallmarks of aging, inflammation is down, stem cells are up, everything that you would think is changing in the right direction, which is great. But going back to my idea of circulating factors, the myokine profile, myokines are muscle secreted cytokines that are coming out of skeletal muscle are actually consistent as that you would see after exercise, which we tend to call exerkines. But believe it or not, I'm actually a neuroscientist. I'm not a muscle expert. So what is actually happening in the brain is what I was really interested in trying to figure out. And so are there any central nervous system benefits to improving proteostasis in skeletal muscle? And so to go back to this cartoon, the three phenotypes we're going to focus on today is a decline in proteostasis, a decline in neurogenesis, and an increase in neuroinflammation. And so we started by looking at the brain of 2-year-old animals. This is quite old for a C57 Black 6. And to look at proteostasis, we just examined the accumulation of lipofuscin, which is an autofluorescent polymer that accumulates in all of our aging tissues, including the brain. And we focused our studies on the hippocampus for multiple reasons. One, it's very easy to see. I call it the banana. You can see it right there, especially for grad students that are not familiar with it. But also because it's an area that's heavily atrophied during both healthy aging and in the context of neurodegenerative diseases. And also it's heavily connected to the ventricles, which receive circulating factors from the blood. So in 2-year-old control individuals, you can see the tip of the banana, if you want to call it that, right there, we're in the hippocampus. There is this accumulation of autofluorescent lipofuscin shown in green. You can see in the zoom in here, all of these essentially inclusions that autofluoresce under the microscope. And when we compared a 2-year-old control to its muscle TFEB expressing littermate, I hope even without the quantification, you can see the difference, but the quantification is right here. There's a significant decrease in the amount of lipofuscin aggregates in the hippocampus of these aging animals. So that was really cool. And all of this work is done by Ian Matthews, another grad student in my lab. So then we wondered, well, is this mediated through neurotrophins, and in particular BDNF? And actually, to our surprise, this is something that I'm still super excited about. There's a significant upregulation of BDNF expression in the hippocampus of 2-year-old individuals that are expressing TFEB in skeletal muscle. And again, this is very consistent to what you see in an exercise model, except these animals are all sedentary. And again, going back to neurogenesis, one of the ways that we've started to look at this phenomenon is by looking at dividing cells, which you can label by injections of EDU. We've also stained them for classical markers of neurogenesis and neurostem cells, such as double cortin. And we have started to quantify their presence and location in the hippocampus. And preliminary data does suggest that there's an increase in the number of these cells that are present in the aging hippocampus in the context of a muscle TFEB expression model. And so this is all great, but they're all hallmarks or benchmarks, as we learned yesterday, that this actually translate into cognitive function. And so we perform a battery of tests, and today, and I'm only going to be telling you about 2 of them. One is the novel object recognition, and I'm just going to move through this in the interest of time, but we do see that our muscle TFEB individuals at 18 months of age, when these animals begin to show deficits, the muscle TFEB expressing animals perform better in the novel object recognition test, as well as the Barnes maze. They escape faster and commit a lot less errors when they escape the open Barnes maze. And so what is happening then in the brain, right? We have all these hallmarks of aging, but what is actually occurring? Well, there's actually significant transcriptional remodeling that's happening. So we submitted the hippocampus RNA lysates from the hippocampus of these animals at 2 different time points, and we actually found that their hippocampi is significantly different than their age-matched littermate controls. There are things that are upregulated, there are things that are downregulated, and so what are the differentially expressed genes and the functional pathways associated with this? Well, really cool things and things that you would expect. And also there appears to be a sex difference. So females are turning on things associated with synaptic plasticity and cognition. So the remodeling of the synapse, both at the vesicle level, at the neurotransmitter level, at the voltage, or the ion channel level. You can see that here. Whereas the males appear to be turning on things that are associated with mitochondria and ribosomal biogenesis. So I don't know what this means yet, but this does suggest that just by modifying or maintaining skeletal muscle proteostasis, we can actually remodel the aging brain. And so again, I don't think we need to tell this audience that all of these features that I've been talking about today are also found in Alzheimer's disease. So we wondered, could this be also an avenue to intervene against neurodegenerative diseases? And so we crossed these animals to a model of tau pathology. These animals, you can read more in the table there, but essentially they develop tau tangles, which is one of the hallmarks of the Alzheimer's disease brain. They also have extensive neuroinflammation. And here I am showing you that we've indeed generated animals that express TFEB and skeletal muscle and have tau in their central nervous system. And so we measured levels of tau accumulation, and that is shown here. This is once again the hippocampus. We have phosphorylated tau shown in white. You can see heavily accumulated in the hippocampus of these individuals. And again, in age-matched littermates that are expressing muscle TFEB, we have significant reductions in both total tau, but also specifically intracellular tau shown here in the insert, which is believed to be the toxic isoform. And we also reduce neuroinflammation. This is an immunostaining of the hippocampus of these individuals, showing you GFAP astrocytes in green and IBA microglia in red. I hope you can appreciate how angry everything looks in this middle panel here. And again, how it looks less angry in the muscle TFEB expressing animals, and the quantifications are shown here. We have significant reductions in the volume of microglia, which is a marker of how angry they are. And also a reduction in the number of astrocytes that infiltrate the area, suggesting we're reducing neuroinflammation. And finally, we've also done medium throughput RNA-seq of the hippocampi of these individuals, if you would, by using the nanostring and counter Alzheimer's disease panel, which collates 700 different genes, collated from human and animal model studies. And we submitted the RNA lysates of 6-month-old animals for this study. And there's too much data here to go through in the next minute, but just suffice it to say that we're also modifying multiple parameters associated with tau pathology and Alzheimer's toxicity, including transcription and splicing, lipid metabolism, and trophic factors. So if you want to learn more about this work, this is actually now in bioRxiv and is under review. So our working hypothesis right now is that we believe that improved muscle proteostasis throughout aging may be a novel central nervous system rejuvenating mechanism. We've improved muscle proteostasis by overexpressing TFEB. We've improved muscle metabolism and preserved muscle health during aging. And we have also believe that TFEB preserves what we like to call the young muscle secretome. And if you want to learn more about that, my superstar master student, Dalton, is actually presenting a poster tonight, so please stop by. But overall, we believe that we are showing improved neuroprotection both during cognitive aging and in the context of age-associated proteinopathies. So with that, I'd like to thank the people that made this work possible. This is my lab, our sources of funding, and I'd be happy to take questions.
Cap it at 2. Yeah, okay, we got 1, 2.
Hi, excellent talk. I had a question. It looked like this was a lifelong model. And so I was curious if you've thought about intervening later in life and if you would still see the same degree of benefit.
Yeah, absolutely. So there is an inducible CRE model that you can turn on with either dox or tamoxifen. I would love to do that. It's part of my next arrow one. But yes, so the idea of a window of intervention, when do I need to turn this on for the effect to still occur? Absolutely.
Thanks.
Hi, I'm Cara from Wisconsin. That was lovely. I was wondering, and sorry if I missed it, when in your TFEB-overexpress mice, did you see any changes in body weight or food intake or anything like that?
Yeah, so we measured everything. So they don't eat more, but their bodies look very different. So they do weigh more. And initially I thought because, oh, the muscles are going to be bigger, and it's not the muscle, it's actually the fat. So this talk was focused on the muscle to brain axis, but as many of these audience knows, everything talks to everything. And so we believe this is a systemic benefit. But yeah, absolutely. There's a metabolic phenotype here that we need to look into.
Awesome. Thank you.